And now I would just like to introduce our speaker of today, Dr. Carlie LaLone from US EPA. Carlie has studied biology at University of Minnesota and then did a PhD at the University of Iowa in the US. But all her life, also from the time that she's been a student for different appointments, she's been working for the US EPA, Environmental Protection Agency. And since 2010, actually, so I believe already for 12 years, she holds a permanent post there, first as a biologist and then moving firmly into bioinformatics, and now she holds a position of the bioinformatics research scientist. And Carlie's main focus of her research is understanding toxic effects on chemicals on all different species which encounter them in the living world. And this she does with the use of sophisticated bioinformatics tools, which she believes are actually not so complicated and could actually be understood by everyone, which is now will demonstrate to us, hopefully, with this webinar. And with this, Carlie, the floor is yours. Please.
Okay, well, thank you so much for the invitation to come and chat with you all today, Ksenia, and those that organized this seminar series. I'm happy to be here. I'm just going to reduce some things so I can see better my slides. Well, yes, so today, I'd like to talk to you all about bioinformatics and particularly applications to challenges in ecotoxicology. And my purpose in presenting to you all is to, both bring you into some of the work that we're doing at the US EPA, but also to let you know that I do think that bioinformatics can be for everyone, particularly interested in species extrapolation. So let me go ahead and move my slides forward here. So I thought it might be a good idea. Many of you may be familiar with bioinformatics already, but just as a broad overview, this science combines mathematics and information science as well as biology to answer biological questions. Typically, it's exploring very, very large data sets of biological data, and this typically also pertains to DNA or protein sequence or amino acid sequences. But what's wonderful about bioinformatics, and it lets the computers do the work for us. Certainly there's interpretation that's necessary of utilizing the tools and the information that's generated, but we can do so much more sophisticated utilizing those types of approaches. And there's been a number of different tools, databases that have been developed to really kind of walk along this sequence to structure to function and being able to utilize such information to make comparisons particularly across species. And these tools have been built in a manner so that most people with basic biological backgrounds can utilize these tools. But one thing that is challenging is, to many people, these are black boxes. They might do something that sounds interesting, but for decision making, for regulatory decision making, for risk assessment, and even for our research, we have to have a strong understanding, a scientific understanding of how these tools are working and why we're utilizing this information and moving from the sequence to structure to function. And so really much of my work has focused on kind of unraveling the complexity and being able to pin together these appropriately developed tools to do a job and take advantage of this type of sequence information, structural information, and functional prediction information for ecotoxicology. And much of the work that has advanced the science of bioinformatics has been from advances in drug discovery and development. So, in drug discovery, there's bioinformatics toolboxes that allow people to take structural information. So say protein structural information that's generated from X-ray crystallography and develop models, computational models that allow for virtual screening of various chemistries. And so a lot of times in drug discovery, you're interested in perhaps humans or companion animals, and you're interested in identifying those chemistries that interact with a particular protein in a particular manner and bind the best, or are likely to interact the best, to be able to move forward with more expensive tiers of testing to understand the efficacy of those drugs. And so there's been these bioinformatic toolbox that have been developed, and certainly with COVID-19, many of these approaches have been advanced significantly, over the last 2 years. And so just to name a few, and we'll talk about this more, there's molecular modeling, molecular docking, virtual screening, which allows you to take hundreds to millions of different types of chemicals and be able to understand how they might interact with a particular structure, protein structure. There's molecular dynamic simulation that basically makes that interaction fluid, so you can see how that would occur in a dynamic fashion. And then there's functional predictions. So for me, my interest is really focused on the use of bioinformatics approaches to understand chemical protein, as well as protein-protein interactions. And the reason for being interested in these types of approaches is I want to be able to utilize this information for extrapolating knowledge across species. And so this is just an overview of what I'll talk about today. So we want to just talk a little bit about what species we rely upon for toxicity testing and why. We'll talk about the consideration of predictive and computational approaches, and why we might want to start thinking about utilization of those, as well as how bioinformatics might be able to help in chemical safety evaluations, particularly thinking about protection of the diversity of species in the environment. I'd like to talk to you about a tool that's available currently and what we're doing to advance that tool, and then how I think that we can begin to incorporate bioinformatics in decision making. So to start off, this is nothing new for this group. We know that chemicals make up the world around us and are certainly very necessary for the modern society that we live in. And this is just a picture of Duluth overlooking Lake Superior. So this is where I live currently and work. And we understand that we have to understand the potential toxicity of these chemicals to both human health and the environment. In the context of the US Environmental Protection Agency, we have a number of legislative mandates where we need to be able to understand toxicity. And so there's chemical safety assessments that require toxicity testing. And this is just an example of some of those that we are interested in being able to generate toxicity information to support regulatory decision making. And we recognize that there's needs for advancing species extrapolation. So certainly for toxicity testing, we know that historically we've utilized whole organism toxicity tests to look at apical endpoints like growth, reproduction, mortality, and more so, we've started to utilize what are being termed kind of this umbrella term of new approach methods. So just as some examples of that, it's typically cell-based or high throughput. So for example, high throughput transcriptomics or high throughput screening assays, such as you might have heard of ToxCast assays. We use the adverse outcome pathway framework to be able to define, the biology, those biological pathways for specific species. But a lot of times, even with these new approach methods, we take advantage of model organisms and use these model organisms as surrogates to be able to understand toxicity across the diversity of species. So regardless of if we use historical toxicity testing or results from new approach methods, even when we're building AOPs, we may have information or data on a single species or a handful of species. And so we want a better understanding of how these other species that are model organisms may act as surrogates. And, just thinking about that, these surrogate species, typically they've been chosen because they're cheaper and they're readily available, or they're easy to maintain. They have short lifespans and rapid life cycles. They don't require a great deal of space and the time consuming care, it's not usually a consideration. And then certainly you're able to control diet and surroundings. And so many times when these species have been chosen for surrogates to use in a laboratory setting, it's because of those types of characteristics as opposed to how good of a surrogate they may be for the diversity of species or even their taxonomic group. And so that's why I'd like to think about species extrapolation and adding additional lines of scientific evidence for why we may utilize surrogate species and be able to extrapolate to the diversity of species in the environment. So, when we talk about species extrapolation, it's really taking what we know about a species or, those handful of species and being able to predict or project the trajectory in other species. So when we're thinking about chemical safety, we're thinking about toxicity. So this is important because we don't have toxicity data for many species. Like I said, there's typically a focal group of organisms that have been selected because of their ease of maintenance in the laboratory. Some, of course, because of their genomes being available. But certainly we just don't have a great deal of information on chemical toxicity across multiple species. And really it's impractical to be able to generate new data for the diversity of species as well. And we all are very familiar that testing resources are limited, but there is an international interest to reduce the use of animals, whole animals, and animals in general in toxicity testing. But on top of that, there's an ever increasing demand to evaluate more chemicals in a timely fashion. And sometimes this includes an expedited fashion. And so we need to be basing our knowledge, our understanding of toxicity on scientifically sound methods for cross-species extrapolation that are transparent and understandable by the researchers as well as decision makers to be able to understand toxicity across the diversity of species, which becomes extremely important in the context of threatened or endangered species as well. So how can we start thinking about the use of bioinformatics in species extrapolation? So the work that really inspired my work was some work done by Lena Gunnarsson. And this was back in 2008. And she had wrote a paper, this is the title, Evolutionary Conservation of Human Drug Targets in Organisms Used for Environmental Risk Assessments. And she really laid out this vision for how we might be able to begin utilizing protein sequence information and thinking about how conservation might be linked to susceptibility or sensitivity of species, and thinking about doing some sequence type comparisons. And this, I think it's just brilliant work and really inspired me to move forward with the work that I'm going to describe to you today. And the idea here is to really begin very simple in the use of bioinformatics and advance as the science begins to advance as well. So what we wanted to be able to do is consider sequence and structural attributes to understand protein conservation across species. So where could we begin? We want to understand species similarities and differences. And so what we did is we were looking for existing and expanding data sources that don't necessarily require the destruction of live organisms to collect samples. So as you know, sequence and structural data is just growing immensely. So there's just new tools and technologies for improved sequencing technologies. There's just large databases and repositories of sequence information that's being curated and made publicly accessible and available. And one source of that data is the National Center for Biotechnology Information Databases. So there's a protein database, and again, it's publicly accessible information. You're also probably familiar with UniProt, but just as an example, NCBI contains over 224 million proteins representing some 117,000 organisms. And I have to say, I check this quite frequently because I present about SeqAPAS often and about this work often. And this protein database, it grows by tens of millions of proteins very rapidly within months. And so we're continuously getting more and more sequence information. There's consortia that focus on sequencing whole genomes for diversity of species. So the promise of utilization of this type of data is continuously expanding, as well as, people are getting better at annotating that type of data as well, which is very, very important for being able to use it for the types of approaches that we're going to talk about. So with proteins, if we're thinking about utilizing proteins for species extrapolation, we want to understand, that these are, you might have heard them as like beads on a string or pearls on a string. Basically, protein sequences are made up of amino acids. These amino acids form a primary amino acid sequence, which is typically the full-length protein sequence that then fold up into secondary structure, alpha helices and beta sheets, and then also fold even further into tertiary structure and quaternary structure. But thinking about proteins, we also understand that there are natural ligands within our bodies that, and all organisms, that allow specific functions, phenotypes, et cetera, within our bodies. But also, a lot of times, chemicals can interact with proteins. And if you understand the adverse outcome pathway framework, so I didn't talk about this a lot prior, but adverse outcome pathway framework considers existing information and helps us bring together biological pathway information that provides us a means to have causal linkages from a molecular initiating event, which is highlighted there in green, where you have a chemical biomolecule interaction. And we collect information that exists to have those causal linkages and weight of evidence moving through levels of biological organization out to an adverse outcome of concern. So whether that's something that we're used to using for regulatory decision making, like growth, reproduction, mortality, we can link these molecular level changes up through the biology to those apical level responses that we're used to making decisions. So this is important because in considering using bioinformatics, particularly in the manner I'm going to describe, we can start thinking about extrapolation of AOPs as well, because chemicals do interact at the molecular initiating event. And like we talked about earlier, protein-protein interactions help us. If we understand conservation there, we can also understand key event conservation. And so we can start looking at similarity across species at the molecular level that helps us predict how well these chemicals or this biology is conserved across species, looking at the functional domains as well as critical individual amino acids and how they specifically interact with these chemicals. So to do this in a transparent, rapid, streamlined manner, we developed the sequence alignment to predict across species susceptibility tool. So this is a web-based online tool. You request an account, everybody gets an account so that you can keep track of your jobs as you're running through them. But the tool was first released in 2016, version 1, and we currently released version 6 this past year. So continuing to evolve the tool as the science evolves, centered around bioinformatics, as well as making the tool more user-friendly, because again, it's important that bioinformatics is for everyone, not just those of us that look at sequence data every day. So for SeqAPAS and trying to make extrapolation from the knowledge of a species to other species, there's a couple of pieces of information that are required, and it depends on how much we know about that chemical protein interaction. So first of all, we have to have knowledge of a sensitive species or a targeted species. So we had talked about pharmaceuticals and pesticides. Many times we understand the protein target, the molecular initiating event that chemical interacts with. And in the context of industrial chemicals and other types of environmental contaminants, ToxCast assays and those types of high throughput assays can help us to identify protein targets. Further, if we're utilizing in vitro assays, and we might want to extrapolate, that knowledge across species as well, we know that a model organism is used in those assays, and we can utilize that model organism for that gene that's being evaluated and extrapolate from that information. that model organism. And then, like we mentioned with AOPs, a lot of times there's an individual species or a handful of species that are used to describe that key event or molecular initiating event. We can also use that information to extrapolate across species from those species where we have data. So what we do is we take the protein for which we know the chemical interacts with in that particular species for which we have knowledge, and we take that chemical molecular target in that targeted species or that known sensitive species, and we compare that protein sequence and structural elements across millions of proteins from thousands of species from those repositories of data that I had mentioned, where we understand and we assume that the greater the similarity between those sequences, the greater likelihood that that chemical can then act on that protein in the other species. And so it provides this line of evidence for chemical susceptibility in the context of thinking about a molecular initiating event, but also we're gathering lines of evidence of conservation if we're thinking about conserved biology in the AOP context. So I'm going to get into more detail, but there's 3 levels of evaluation for SeqAPAS. So you can take any species as your starting species. So if you're looking at a pesticide, maybe you have an insect that's known to be sensitive, or a plant species. If you're looking at a herbicide, typically there's a lot of interest in mammalian species and perhaps thinking about pharmaceuticals. So it depends on what your problem formulation is as you go into utilizing a SeqAPAS tool, but you would choose that query sequence in SeqAPAS to align with the known sensitive species for the chemical of interest, or like we talked about, the in vitro assay model organism, or the species that was used to develop the adverse outcome pathway framework. Once we have that information, we submit it to level 1 and we can compare primary amino acid sequence alignments. We can also take it further if we have information, say, on functional domains. So this could be like a ligand binding domain in the context of a nuclear receptor or some catalytic unit, if we're thinking about enzymes. And then further, if there's information in the literature, perhaps from X-ray crystallography data or cytoric mutagenesis data or field resistance, if we're thinking about pesticides, we can identify critical amino acids. So these are amino acids within the sequence that are directly interacting with our chemical of interest or the protein of interest, if we're thinking about protein-protein interactions. It could be amino acids that are important for maintaining the function, or the structure of that protein upon binding. But basically, we can, based on how much knowledge we have, we can gather these different lines of evidence towards conservation to make a prediction of susceptibility or understand conservation across the diversity of species. So to break that down, like I mentioned, Miguel was a contractor that worked for us that developed this nifty visualization that describes SeqAPAS. And like I mentioned, we take advantage of NCBI tools and databases to be able to generate the SeqAPAS tool. So again, it's about being transparent and bringing together the information to make these processes streamlined. So in this example that I'm demonstrating here, as a visualization for SeqAPAS, we're utilizing a human protein target. So let's just say this is the human nuclear receptor, maybe an estrogen receptor, and we're trying to understand what other species are likely to be susceptible to a chemical that we know interacts with the estrogen receptor. So we can start comparing sequences at the full primary amino acid sequence, level 1, and we'll get an understanding of whether or not that sequence is conserved, yes, or not conserved. And so we go through each species. The tool automatically, of course, makes these types of comparisons, and we get these yes or no answers of conserved or not conserved based on percent similarity. And so the way that that's done is by looking for ortholog candidates. So orthologs are sequences that have diverged from a speciation event and are more likely to maintain similar function. And so we use computational methods to identify ortholog candidates that are, again, we're looking for that potential for functional similarity. And so the tool automatically uses a method called reciprocal best hit blast to identify those likely ortholog candidates. And once we gather that information of ortholog candidates, yes or no, we can generate information to set a susceptibility cutoff in the percent similarity data. So this is a little bit more detail, but I think it's important to just touch on here. So basically, this is just a simplification of the output from a SeqAPAS evaluation, where we have the different species, their common names listed here. We have the ortholog candidate, whether it was identified as an ortholog candidate, yes or no, the percent similarity, so comparing those sequences one at a time, you get a percent similarity to the query sequence. In this case, it was the human at 100%. And what we do is we look in the data. So we'd have hundreds or thousands of sequences and data, and we'd say, what is the lowest percent similarity where we're still identifying ortholog candidates to be able to make this assumption of a more likely to have similar function there. And that's where we set our susceptibility cutoff across the whole data set. So those sequences that are above the cutoff are more likely to be susceptible or more likely to be conserved, whereas those below the cutoff are less likely to be conserved. And so what we can do then is we can screen hundreds to thousands of species and have predictions of susceptible yes or susceptible no. We can then move on to level 2. And again, here we're looking for functional domains across the species. So every hit species that we had identified to align in our example here with humans, we then also take that further to look for conservation of functional domains. So the user will select the functional domain of interest. So again, you have to have some knowledge about that chemical protein interaction and what's important for your protein target, which is typically information that's available in the literature. And then we have pieces of the tool that helps to guide you to those functional domains of interest. So again, here at this level, we get a prediction of susceptible yes or susceptible no. And again, going through those hundreds or thousands of species very rapidly. The next level, like I mentioned, is if there's information available on critical individual amino acids, we can look specifically at those important amino acids that are directly interacting with the chemical, for example, or maintaining the function of that or the structure of that particular protein. And we can look and understand how well conserved are those individual amino acids across species. And just an example of this, so you might have with the aryl hydrocarbon receptor, it's been identified in avian species that specific amino acids within the ligand binding domain are important for predicting avian toxicity. So that's just one example of how this has been shown in the literature, where you can identify these individual amino acids that are important for understanding chemical toxicity across species. Of course, there's multiple lines of evidence like that in the literature, thinking about pesticides and mutations that cause resistance, for example. So once we've collected our level 1 through level 2 or through level 3 data, if we have that detailed knowledge, we're adding lines of evidence towards conservation of that target. And we actually add taxonomic resolution to our susceptibility predictions. So what do I mean by that? So at level 1, we're making predictions across the full protein sequence. And so likely we can get predictions across vertebrates, invertebrates, plants. But as we move down to looking at critical individual amino acids, we can start getting species-specific predictions because we're looking specifically at a particular chemical protein interaction that we have knowledge about. So then we get very specific information on whether or not a species would be predicted susceptible or not. And so I want to be really clear on what the tool provides. It provides transparency in the data. So there's data tables that are output that have all the different metrics that you're comparing to be able to generate these susceptibility predictions, as well as links to NCBI and some of the backend tools that are being used so that you can look at specifically the sequences that you're making your predictions using the sequences from NCBI. But the data itself also provides you with a yes or a no, susceptible or not, conserved or not, that you can use for extrapolation across the diversity of species. What SeqAPAS does not predict is the degree of sensitivity or susceptibility. We understand that thinking about the factors that go into species sensitivity, there's obviously toxicokinetics, exposure, dose, ADME, all of that information is so important, as well as life stage, life history. Those are considerations that other types of approaches and other tools could bring to this evaluation if you're thinking about weight of evidence or lines of evidence to support conservation across species for extrapolation. SeqAPAS in of itself asks a very specific question of whether or not that protein target is available in another species for that chemical to act upon, yes or no, susceptible or not. So, it's very specific in what it does. So some of the strengths of SeqAPAS is it's publicly available to everybody. And I'd encourage people, if you have challenges or interests in extrapolating knowledge across species, you should give the tool a try. I think you'd find it as a valuable resource, because we've tried to streamline it in a manner that would make it useful for everybody that has a basic biological background to utilize this tool for a meaningful extrapolation that provides evidence for conservation for hundreds to thousands of species very rapidly. And it takes advantage of well-established tools and databases from the National Institutes of Health within the US government that maintains these types of tools. The tool is meant to take advantage of many different types of bioinformatics approaches, databases, and put it in one interface that helps walk the user through this evaluation specific for species extrapolation. When we've developed the tool, we find it very important to generate case examples to demonstrate applications for decision-making, how this can be used. So I'm going to talk a little bit about that in the coming slides. We have a user's guide as well as help menus throughout the tool. And one thing that's very important, I think, is paying attention to the science. So as bioinformatics approaches evolve and become more user-friendly, where we can automate or semi-automate those approaches for thinking about species at the molecular level and conservation of targets, we want to be able to advance the SeqAPAS tool. And so most importantly, I think, is demonstrating application of SeqAPAS. And these pictures are meant to point out the different types of evaluations that we've done previously. So for the case studies that we've generated, we've utilized the tool to extrapolate AOP, adverse outcome pathway knowledge across species. And this is very important for defining the taxonomic domain of applicability. And of course, for EPA, we're very interested in pollinator health. So we are extrapolating knowledge from APIS and non-APIS species. And I'm going to get into more detail on that particular example in the coming slides. We've also published work on extrapolating high throughput screening data, focusing on endocrine targets specifically, such as estrogen receptor alpha, androgen receptor, enzymes involved in steroidogenesis, as well as the thyroid axis, so proteins involved in the thyroid axis. So you may have heard of the ToxCast suite of assays. We've evaluated all of those targets using SeqAPAS and provided the data through the CompTox chemicals dashboard, as well as through a publication in 2018 that provides that data. I think that's really important to demonstrate its use for extrapolating high throughput knowledge. So typically we're relying on, again, model organisms. We want to understand how broadly can we anticipate extrapolating that information to other species. We've used it just for predicting relative intrinsic susceptibility to pesticides in the context of the Endangered Species Act. So where we're unable to test, for example, species like orca whale, or we just don't have toxicity data as we wouldn't for a majority of threatened and endangered species. But a lot of times there is growing genomic information as well as protein information that we can utilize for predicting susceptibility. Most recently, we've been demonstrating the use of SeqAPAS for derivation of aquatic life criteria in the US. And then further, we've utilized the tool for thinking about bioaccumulation potential for perfluoroalkyl substances. per and polyfluoroalkyl substances, and other chemicals of concern. And then we've demonstrated how the tool can be used for generating research hypotheses, as well as for prioritization strategies. So I can't go through all of these examples, but I'm briefly going to go through one, like I mentioned, thinking about the taxonomic domain of applicability for AOPs. So back in 2017, together with our program office partners from the Office of Pesticide Programs, we developed an adverse outcome pathway network that considers both chemical and non-chemical stressors and activation of the nicotinic acetylcholine receptor, leading to colony death and failure for Apis mellifera, which is the honeybee species. And so we generated this AOP network, which is also in the AOP wiki. We published the work, generating weight of evidence as well, taking into account biological plausibility and empirical support for the AOP. And we did this to both define knowledge gaps, as well as understand the nodes within the network that can be impacted by multiple stressors in a way to assist in the development of mitigation strategies and being able to protect important pollinator species. But one of the key aspects is, okay, so we generated this knowledge specifically for thinking about pollinators and more specifically thinking about honeybees. And much of the data that we collected, of course, was also from the honeybee. And within this network, there was 6 AOPs. And what I want to talk about is defining the taxonomic domain of applicability across an AOP. So to generate a case example, I want to simplify what we're going to talk about. So I pulled out a specific adverse outcome pathway here, again, linking nicotinic acetylcholine receptor activation to colony death and failure. And so how can we use this AOP to define the taxonomic relevance? So you might be familiar, of course, with decision trees. And initially, if we're thinking across the whole diversity of species, there's certainly different taxonomic groups that could be, fall in line with this AOP because, perhaps they live in a colony, they use foraging methods similar to a bee, but there's a lot of taxonomic groups that would obviously fall out if you used a decision tree to walk through the AOP. And so, this is a way to also gather biologically plausible knowledge that this, either element of the AOP or AOP overall would likely be conserved across taxonomic groups. So you can use a decision summary, a decision tree to summarize those species that are those taxonomic groups that are likely to be impacted at these different levels of biology. And then we can also take empirical evidence for the taxonomic domain of applicability. So as I mentioned, for this AOP, you'll see that for on the right-hand side of these boxes, there's A species or up to 3 B species that were used to define these different events. And that's because that was the empirical data that was available. So how can we expand this more broadly? We have empirical data, there's some knowledge of biological plausibility, but we really want scientific evidence that helps us to be able to extrapolate to other species. And so this is a pretty busy diagram, but what we can do is we can look for ways to utilize bioinformatics and specifically the SeqAPAS tool to be able to broaden that taxonomic domain of applicability, focusing on the plausibility, the biological plausibility for the taxonomic domain. So if we look through this AOP, there's 4 key events, including the molecular initiating event, that are proteins are essential for the description of that key event. And so thinking about utilization of empirical evidence, the species that were used to develop the AOP, we have those species listed, but we can then take those species such as the Apis mellifera, and we can extrapolate from the nicotinic acetylcholine receptor and other proteins in this biological pathway to all other species and understand whether or not that biology is likely to be conserved. So if we do that, if we extrapolate from the key event, from the empirical data that exists for the molecular initiating event and these various key events, we then get a list of species from SeqAPAS of the conserved species. So that's the biologically plausible species for that key event. Well, then if we're trying to define the key event relationship, which provides the causal linkage from one upstream event to the downstream event, we can look for the narrowest point between the lists of species from the empirical data and the bioinformatics data to define the biologically plausible taxonomic domain of applicability for the key event relationship. And then further, if we're trying to define the taxonomic domain of applicability for the entire adverse outcome pathway, we can then take those nodes from the key event relationship and understand the common species between all key event relationships. And that would help us to define the plausible taxonomic domain of applicability for the full adverse outcome pathway framework. So I am going to SeqAPAS Europe and Copenhagen this month, and this presentation specific to this example will be expanded much more significantly on Wednesday. But just to give an overview of the results that we generated from this study, this is actually a master's student of mine, Marissa Jensen, that pulled this information together and has a manuscript that we're submitting actually later today to describe this. Here then, using that approach that I had mentioned, using SeqAPAS, and specifically now, as I mentioned, SeqAPAS provides results across hundreds of species, but focusing in on specifically a question looking at APIS versus non-APIS species and expansion of the knowledge there. We simplified this example and looked only at bee species. So we extrapolated to 17 bee species from Apis mellifera for a number of these key events. Here we only have a couple of species because there's no proteins involved in those specific key events. Out here, key event 5, there were proteins involved in the role change for the case within bee species. So we were able to extrapolate to additional species here. And then, as you can see, we followed that approach to be able to define those species that are likely to follow the same biology. Whereas you'd envision that other species that aren't, so we have other bee species identified here. However, if you think about it, there's some species that use colony structures. There's solitary bees. Those types of bees that don't use similar foraging strategies would also fall away from this particular AOP at the later events, though the early key events may be conserved. So if we think about across the whole AOP, it's likely that Apis mellifera, which is the honeybee, and other eusocial, meaning that they're in these types of colonies that work together, Apis species would be included in this particular AOP. So what we were able to do, utilizing both bioinformatics and empirical evidence for the adverse outcome pathway, expand that taxonomic domain of applicability much further to in this bee-specific focus to 17 bee species from the 1 to 3 species that were available for this AOP. And I think that's really important for utility of the AOP and understanding how broadly these types of key events can be extrapolated. Okay, so that was an example of how the tool can be used. There's certainly multiple others that we've published on, but now what I just want to briefly touch on before we're done with time here is where we're moving with the SeqAPAS tool. So like I said, we started with sequence and very simple and started bringing in understanding of different elements of structure. So thinking about important amino acids that are interacting with the chemicals. So we always want to be able to look several steps ahead to be able to take advantage of these approaches. And we talked about earlier the types of advances that are being made in drug discovery, and particularly with COVID-19 and the advances that have been improved, the bioinformatics approaches that have been improved through efforts in identifying a vaccine. So in trying to move forward with SeqAPAS, we want to start considering the use of this bioinformatics toolbox and comparing species and getting to the point where we can model structures as well as utilize the models for comparing across structures and generating additional lines of evidence for conservation of structures, and then have those structures available for molecular docking and some of these advanced modeling techniques like virtual screening and molecular dynamic simulations. So in thinking about how can we apply these approaches to species extrapolation, we can start thinking about generating models for many species and exploring docking with multiple structures. And that's exactly what we've been doing over the last year so that we can move towards a virtual screening of like thousands or millions or even billions of chemicals computationally because of the power of the computer being able to generate these data and our ability to interpret these data in a streamlined fashion. So what we've been doing is moving the tool, advancing the tool towards being able to think about comparisons of protein structure. And I'm not going to get into significant detail here, but we've utilized output from SeqAPAS and tools that exist to be able to generate models across the quality protein sequences that we have, and then utilize those models for comparing a query model structure to all of the hit model structures that we're able to generate, and then being able to eventually thinking about how we can advance the tool beyond the yes or no prediction, and potentially at some point moving into more advanced methods, generate predicted binding affinities. So that's where the tool is moving, because there's an ability to start thinking in that direction because of advances that have been made in bioinformatics. And we'll be having a paper that should be submitted later this summer that highlights how you can utilize that type of a pipeline to start looking at protein structure for species extrapolation. But what I want to touch on now is just recognizing that many of us are developing tools that help us start thinking about conservation of sequence, structure, and function, and biological pathways and AOPs. And so in an effort to try and bring together those that are developing these types of approaches, these types of tools, as well as champions that want to be able to utilize this type of information for decision making and recognize the need to be able to take advantage of existing knowledge, we wanted to really bring together a group of international, a focus group, a consortium to advance cross-species extrapolation and regulation. And so a number of us that are very interested in this area of science and advancing in decision-making have gotten together and developed an international consortium specifically to advance cross-species extrapolation and regulation. And we have a number of different objectives that are listed here, but just of interest, defining that global regulatory landscape and the need for these types of approaches, but really being able to demonstrate in a global group that's working together, trying to bring together the state of the science and bioinformatics and demonstrate in case examples how we can utilize these tools and make them available, and what types of criteria, what types of guidance is needed to be able to bring this scientific, these types of approaches in computational methods to our decision makers. So we want to have both the regulatory community as well as those scientists that are heavily engaged in species extrapolation, and particularly bioinformatics as a starting point, together. And so there's this group that we're working on bringing together. Our first actual team meeting is June 22nd. So if you are interested in being involved in this type of discussion, or have some tools that you're developing that you think would be useful, I'd invite you to just shoot myself or Jeff Hodges from Unilever an email, or any of the steering committee members here, and we'd be happy to include you in that meeting and get you integrated with the consortium that is kind of the umbrella organization has been CTEC, so the Society of Environmental Toxicology and Chemistry, that's supporting this consortium, as well as a variety of specialty sections within the Society of Toxicology that are also supporting this consortium. Finally, my acknowledgements slide: Marissa Jensen, all of the taxonomic domain of applicability for AOPs relevant to APIS and non-APIS species, that is her work. She's a master's student working with me, and all the others that provide great research and case examples to demonstrate the utility of SeqAPAS and other bioinformatics approaches are here, as well as the GDIT team that they're the computational experts that actually develop, create the SeqAPAS tool. So My email is listed there, so if we don't get a chance to answer all of your questions, please feel free to reach out. I hope I do see some of you at CTAC Europe, and I'd invite you to go to the SeqAPAS URL and get yourself an account and check it out. You can't break it, so might as well try it and see how it would work for you and your research or decision-making needs.